A'udhu billahi minash shaitanir rajim. Bismillahir Rahmanir Rahim. Assalamu alaikum, dear students. Today we will discuss proteins, and the class is globular and fibrous proteins. You will learn structural classification of proteins. You will differentiate between globular and fibrous proteins. And in detail, we will discuss structure and functions of myoglobin, which is a globular protein, while among the fibrous proteins, we will discuss in detail about collagen, elastin, and keratin, and also the clinical disorders associated with these proteins. Proteins can be defined as a group of high molecular weight nitrogen containing organic compounds. They have the complex shape and composition. The molecular subunits of the proteins are polypeptides, which are made up of different amino acid linked together by the peptide linkage to form long chains of polypeptides. So amino acids, they make the polypeptides, and polypeptides, they make the proteins. Here is a guide of 20 common amino acids. It is good for ready learning, and the emphasis on the side chains of the different amino acids so that you can understand the structural composition difference, and you can understand how sort of reactions they can undergo. It is only for the ready reference which will help you in understanding of structure and function of fibrous and globular proteins. Proteins can be classified in different ways, but on the basis of structure, they can be divided into two: globular proteins and fibrous proteins. Globular proteins, they have axial ratio, which is less than 10, and usually it is not over 3, while the fibrous proteins, they have axial ratio 10 or more. You can see the structure of fibrous protein, which is fibrous in nature, long, thin, filamentous in structure, while the globular proteins, they have globular or circular ball-like shapes and structure on which they are classified as globular proteins. Globular proteins are folded into spherical or ovoid shapes. They are usually water soluble, and they usually have roles in metabolic reactions. Examples are hemoglobin, myoglobin, enzymes, globulins, and albumin. There are various other proteins which are also globular proteins, have various functions, but the important examples of the globular proteins are these. While the fibrous proteins, they have thread-like fibrils or long fibers, long twisted polypeptide chains, cross linkages at intervals forming long fibers or sheets, usually insoluble in water, and many have structural roles, and they are generally found in extracellular matrix. Examples are collagen, elastin, keratin, tubulin, actin and myosin, and fibronectin. This is a table showing the major differences between fibrous and globular proteins. Fibrous proteins are insoluble, while globular proteins are soluble in water. Globular proteins are involved in metabolic functions, while the fibrous proteins, they form the structural elements. They have long, narrow shape, round or spherical shape, helical or sheet structure, while globular proteins are folded bile-like structure. The fibrous proteins generally have the only secondary structure or secondary level of the structure, while in globular proteins tertiary structure is there, and some proteins even have quaternary structure like uh, hemoglobin. They have repeated amino acid sequence, while the globular proteins, they consist of an irregular amino acid sequence depending upon the primary structure of that protein. They are insoluble in water, acids, and bases, while globular are soluble in water, acids, and bases. They have strong intermolecular interactions so that they provide the fibrous proteins, give you the strength, and they are strong in structure, while the globular proteins are weaker intermolecular interactions they have, and they are weaker in their structure, less sensitive to the changes in temperature and pH, while they are sensitive to the changes in temperature and pH. They have globular proteins, they can be denatured by the change in temperature or pH. Their function can be altered. Their fibrous proteins are involved in forming structures, while they are involved in metabolic. Globular proteins are involved in metabolic reactions. Among the globular proteins, two most important, hemoglobin and myoglobin, we will discuss. The hemoglobin is taught to you in detail. The synthesis of heme, structure and function of hemoglobin, and even the oxygen carrying capacity and phenomena by the hemoglobin have also been taught to you. Here only few points for your recall, that it is an example of globular protein, reddish purple oxygen carrying pigment of heme they contain, and the hemoglobin is made up of 4 polypeptide chains, 2 identical alpha and 2 identical beta chains. They are nearly spherical in shape, and hydrophobic side chains made up of hydrophobic amino acids. They are pointed inwards by the hydrophobic interaction, while the hydrophilic side chains made up of hydrophilic amino acids, they maintain solubility and point outside of the globin molecule. Each polypeptide chain contains of a heme group, which is a prosthetic group tightly bound with the globin molecule. Globin molecule is a globular protein. Heme group is a prosthetic group. It is an important permeable part of the protein molecule, which is not made up from amino acid and cannot be separated easily, and it is combined with the polypeptide chains. Heme group has iron, and iron combines with oxygen to carry it and transport the oxygen. One hemoglobin molecule having 4 heme groups, they can carry 4 oxygen molecules. Color is bright red when combined with oxygen. Myoglobin is a monomeric hemoprotein present in red muscles. If you remember, the hemoglobin is tetrameric hemoprotein. Myoglobin consists of a single peptide chain similar to beta chain of hemoglobin, and this single polypeptide chain is having 153 amino acids with molecular weight of 17,000 Dalton. And you know the 1 Dalton is equal to approximately the mass of a hydrogen atom, or 1/12 of a mass of carbon atom. The high proportion of the polypeptide chain, up to 75 to 80%, is folded into 8 right-handed alpha helices. These alpha helices, they are labeled from A to H. A, B, C, D, E, F, G, and H. The surface of myoglobin is hydrophilic, while the interior is hydrophobic, similar to globin chain of the hemoglobin. The alpha helix is E and alpha helix F. E at position number 7 and F at the position of 8. They have the histidine amino acid, which lie close to the heme iron, where they function in oxygen binding. Here is the oxygen binding presentation of myoglobin. Proximal histidine of helix F helix at position number 8 is bound with the iron of the heme, while the histidine, distal histidine at helix E position number 7 is also present there, having the helping in the stabilizing of the binding of the oxygen with the heme. Functions of myoglobin. Myoglobin binds one oxygen molecule reversibly because it is oxygenation, not the oxidation. The oxygen dissociation curve for myoglobin has a hyperbolic shape, while in hemoglobin it is sigmoidal in shape. This hyperbolic shape of oxygen carried by the myoglobin reflects higher oxygen affinity at all PO2s. When the partial pressure of oxygen is low or even it is high, in both the conditions, the oxygen affinity for the myoglobin is more and the binding or association of oxygen to the myoglobin takes place. It receives oxygen and transports, and myoglobin also stores oxygen. And when it stores oxygen, this oxygen can be used in case of, for example, in case of severe exercise, when there is oxygen deprivation, for use in the muscle mitochondria for aerobic synthesis of ATPs, because the muscles, they need energy in the form of ATPs. Common conditions leading to myoglobinemia or myoglobinuria. In accidents resulting massive crash injury, myoglobin is released from damaged muscle fibers. When the muscle fibers, they are damaged, that condition clinically is generally called rhabdomyolysis, the breakdown of the skeletal muscles. And this presence of myoglobin colors the urine dark red. Because of the presence of myoglobin cast are the tubular or cylindrical structures which are stuck up in the kidneys, and there may be acute renal tubular necrosis, and there is acute renal shutdown, which is an emergency and requires dialysis. Gas gangrene, tetanus may also cause mild myoglobinuria. Gas gangrene is the necrosis of the part of the body due to bacterial infection. Tetanus is the spasm or stiffness of the muscles caused by the microorganisms which cause mild myoglobinuria. There are various myopathies, could be genetic or acquired, which may cause myoglobinuria, but they are found with muscular dystrophies. Before starting fibrous proteins, it is important to understand the structural functional relationship of proteins. For example, myoglobin and hemoglobin, they have different structures. When they have different structures, the hemoglobin is tetrameric while myoglobin is monomeric. Hemoglobin carries 4 molecules of oxygen while myoglobin carries 1. The oxygen dissociation curve is hyperbolic in case of myoglobin and it is sigmoidal in case of hemoglobin. At higher partial pressure of oxygen, there is complete saturation of hemoglobin, while the myoglobin can be saturated even at low partial pressures of oxygen in the blood, and even in case of severe exercise. Hemoglobin transports the oxygen while the myoglobin stores as well as transport the oxygen. You appreciate that this is the difference of functions just because of that change in structure. So structural and functional relationship is very much important, which is also would be seen in case of fibrous proteins. Now we start with the fibrous proteins. The fibrous proteins, they are most commonly synthesized in fibroblasts of connective tissue, and they are found in extracellular matrix performing various functions. The various functions performed by the fibrous proteins, the main functions are they provide rigid and elastic structure, they help in addition of the cells, molecular filtration, and tissue repair of the body in case of damage. In the class of fibrous proteins, the most abundant protein found in the body is collagen. It is most abundant protein in human body, and a typical collagen molecule is a long rigid structure made up of 3 chains, 3 alpha chains, alpha 1, alpha 2, alpha 3. 3 alpha chains are wound around one another in a rope-like triple helix or super helix. This is the presentation of a typical collagen molecule with alpha chains. Let us go for the properties. The collagens are mostly abundantly found in the body and has a family of about 25 different types. Type 1, type 2, type 3, and so on. There are most abundant proteins in mammals, consisting of about 25% of total proteins. The main function of collagen molecule is to provide a structural support to the tissues. It is the organization of most abundant and most common types of collagens. Type 1, type 2, type 3, 4, 5, 7, 9, and 12. These types, they are grouped into 3 main groups: fibril forming, network forming, and associated with fibrils. In skin, bone, tendon, blood vessels, and cornea, type 1 collagen is abundant. Cartilage, intervertebral disc, vitreous, body of the eye, type 2 is present. In blood vessels and fetal skin, type 3 is also present. The collagen types 6, 4, and 7, they form networks found in basement membrane on which the epithelial cells are attached, and they are also found beneath the stratified squamous epithelium where there is no basement membrane only the collagen fibers, they provide the site of attachment for the epithelial cells, because here they need the stretch, for example in the bladder, urinary bladder. Third group is fibril associated, type 9 in cartilage and type 12 in tendons, ligaments, some other tissues. In this slide, you see the arrangement of collagen fibers in various tissues of our body. In tendons, there are parallel bundles of collagen. In skin, sheets of fibril are present. In bones, fibers are arranged at an angle to each other so as to resist mechanical shear from any direction. Cartilages have no distinct arrangement, while in cornea, the planar sheets are stacked crossways so as to minimize light scattering in the daylight, and when we see in the light, there could be scattering which is prevented when they pass through the cornea because of the presence of the planar sheets stacked crossways. Structure of collagen. A typical collagen molecule is a long and rigid structure made up of 3 alpha chains. 3 alpha chains or 3 polypeptides, they are wound and make one long and rigid molecule of collagen. Each polypeptide subunit or alpha chain, each alpha chain is twisted left-handedly to make the alpha chain. When 3 alpha chains or 3 polypeptide chains, they wound right-handedly. So this opposite twisting of the alpha helices or alpha chains, they provide great strength to the collagen molecule. The basic unit of collagen is tropocollagen, which is found extracellularly. Intracellularly, in various steps, a molecule of procollagen is formed, which is water soluble, and outside the cell, when procollagen is secreted, an insoluble molecule of tropocollagen, which is the basic unit of collagen, is formed. As we compare with the alpha helical structure of the other proteins, the collagen alpha chain is much more extended, having 3.3 residues per turn. This extended structure of alpha chain provides the flexibility to the collagen molecule. The amino acid composition of collagens, as they are rich in glycine and proline, but some amino acids of lysine, they are also present. The collagens are unusual because they contain hydroxyproline and hydroxylysine, which are not generally found in other proteins. Every third residue or third amino acid is glycine, and in X generally proline is there. In Y there could be hydroxyproline or hydroxylysine. So glycine, proline, and hydroxyproline or hydroxylysine, these three amino acids, they are found in the molecule of the collagen. Functional purpose of amino acids. The amino acids glycine, proline, and hydroxyproline and lysine, they have purpose. They make the structure of collagen, and which provides the functional properties of collagen molecules by virtue of these amino acids, which form the primary structure of the alpha chains. Glycine being the smallest amino acid, helps to pack the alpha chains tightly together to form the final collagen superhelix or triple helix. While proline is the unique amino acid, and it is also known as amino acid because it has 5-membered strong structure having the secondary amino group, not the primary one, and is a kink molecule and helps in the kinking and stabilizes the helical conformation each chain. Here in the diagram, glycine the smallest, helping in the packing, hydroxyproline providing the kinking and strength to the molecule. Proline is also providing the kinking, and a strong molecule or triple helix is formed. In this diagram, 3 alpha chains A, B, C are there. The glycine amino acids, they are tightly packed together and help in the formation of a triple alpha helix rope of the collagen molecule. The X is proline, Y could be the proline, hydroxyproline or hydroxylysine, which has got important functions in the molecule of the collagen, which we will discuss in the next slides. Hydroxyproline and hydroxylysine, they are unique. They are not present in most other proteins because they fulfill the specific structural function when they are present in the collagen molecules. The collagen molecules are in the form of procollagen synthesized inside the cell. Inside the cell, transcription takes place, then translation and synthesis of proteins in ribosomes. When the alpha chains are synthesized in the ribosomes, there is post-translational modification inside the cell. That is, after completion of the translation, change in the polypeptide chain occurs, and some of the proline and lysine residues are hydroxylated to make the hydroxyproline and hydroxylysine, which have got the unique role in the structure and function of the collagen fiber. Here is a proline residue, could be the lysine residue. The enzyme is prolyl hydroxylase or lysyl hydroxylase, and molecular oxygen is utilized, and the hydroxylation of this protein or this amino acid takes place in the pro-alpha chain. This hydroxylation of the proline molecule is very much important in understanding of the structural and functional relationship of the hydroxyproline and hydroxylysine, whatever may be the case. You see the ascorbate, which is the active form of vitamin C, is needed as a reducing agent. And that's why in deficiency of ascorbic acid or vitamin C, this hydroxylation of proline and lysine residue is minimum, causing the poor structural formation of collagen fibers by virtue of less hydroxyproline and less hydroxylysine formation. Purpose of hydroxyproline. Hydroxyproline provides the hydrogen bonding formation between the chain. 3 alpha chains, making the helical structure of collagen molecule to form the superhelix is by virtue of hydrogen bonding of the hydroxyproline. If hydroxyproline is deficient, the abnormal collagen molecule is denatured and cannot withstand the temperature beyond 20 degrees centigrade, while normal collagen rich in hydroxyproline can withstand the temperature beyond 40 degrees centigrade and is denatured after 60 degrees centigrade. So the hydroxyproline provides hydrogen bonding between the chains so that three alpha chains can make a rope-like strong molecule of collagen. Hydroxylysine also performs important function. As you see, glycine is the smallest amino acid providing the packing, proline providing the kinking, hydroxyproline providing the hydrogen bonding. The hydroxylysine is also provides the hydrogen bonding as well as covalent cross links, which are called aldol cross links of collagen fibrils. Some of the lysine residues, they are oxidized to their aldehydic derivatives, which form aldol cross links. These aldol cross links, they are covalent links and very strong in nature, providing greater strength. Hydroxylysine also provides the site for the attachment of carbohydrates, glucose and galactose, making the collagen a glycoprotein. These covalent cross-linkings, they have importance. The covalent cross-linkage takes place between the triple helical units or superhelix and inside the superhelix, both within and between the triple helical units. These cross-links stabilize the side-by-side packing of collagen molecules and generate a strong fibril. If inhibited, the tensile strength of fibril is drastically reduced. The collagenous tissue becomes fragile and the structures like skin, tendons and blood vessels tend to tear. The amount of cross-linking in the tissue increases with age, both in humans and animals, and that is the reason meat from older animal is tougher than the meat from younger animals. Formation of mature collagen fibers. The synthesis and formation of the collagen fiber is well illustrated in the lipid cord. You can see the diagram and can recall and understand the formation of collagen fibers. But few steps briefly I can describe here. In the cell, inside the cell, there is transcription, translation and post-translational modification and hydroxylation of the proline and lysine, and alpha chain is formed. Then there is a formation of triple helix and a soluble molecule of procollagen, which is triple helical, is synthesized inside the cell. They are soluble. Procollagen is soluble because of the side polypeptide chains, which are water soluble, which have to be broken down. So the procollagen is released from the cell. The side chains, soluble chains, they are broken down by the procollagen peptidases, and a basic unit of collagen fiber, tropocollagen, is formed. This tropocollagen molecule is around 300 nanometer in length. They make the collagen fibrils by virtue of bondings and covalent linkages. And these collagen fibrils, they develop also cross linkages, and a mature collagen fiber is formed. A mature collagen fiber can be in the form of fascicles or bundles, as seen in the nerve or muscle fiber. Scurvy is the disease of connective tissues by virtue of deficiency of vitamin C. It should be taught along with the vitamin C. But here it is worth to study the scurvy to understand the scurvy and connective tissue disorder at the molecular level. Scurvy is the disease caused by dietary deficiency of vitamin C, which results in the defective connective tissue formation. You know the vitamin C is required for hydroxylation reaction of proline and lysine to make hydroxyproline and hydroxylysine. These defective pro-alpha chains in which the hydroxylation should take place, but because of the deficiency of vitamin C, hydroxylation is less or poor. So poor alpha chains are formed, which cannot make a stable triple helix. And these pro-alpha chains, they are degraded. The blood vessels become extremely fragile and may bleed. You can see the gums, bleeding gums, and there is hemorrhage and swollen gums with loss of teeth if untreated. These swollen gums or gingivitis can result in the loss of teeth if the deficiency is prolonged, usually seen in the sea voyagers in the history. They used to have these conditions, and later on they found that because it is deficiency of vitamin C, which they never used to take during their sea voyages. And there is also bruises found in the limbs because of capillary fragility and bleeding, and there is delayed wound repair and healing. The role of vitamin C, you see here, the formation of hydroxylated proline or lysine, which are not incorporated into alpha chains and results in the poor formation of collagen fibers. Now we'll discuss the collagenopathies or clinical disorders associated with collagen. There are more than 1,000 mutations that have been identified in 22 genes for 12 of the collagen types. There are more than 25 types of collagen, but 12 are more important. One is Ehlers-Danlos syndrome, EDS, which is heterogeneous group of generalized connective tissue disorders. Clinically, the gene for type 3 collagen is defective, or also there is defect in gene of the collagen type 1. But most importantly, type 3 collagen, mainly found in blood vessels, it is potentially lethal, creating lethal vascular problems. When the blood vessels or their arterial walls, they are weak, they bulge out and they make the aneurysms. So bulging of an arterial wall is aneurysm. These aneurysms do not cause any clinical problem, but when they rupture because of the weakness, they could be lethal and lethal bleeding if timely operated otherwise. If the patients show defect in collagen type 1 fibrils, there is a stretchy skin and loose or flexible joints, which can be seen in Ehlers-Danlos syndrome. In the collagenopathies, second condition is osteogenesis imperfecta, OI, which is also a syndrome. Structurally abnormal chains, they prevent folding of the proteins into a triple helical conformation. Most patients with severe disease have mutation in the gene for type 1 collagen. This is the lethal form, or type 2. Type 2 osteogenesis imperfecta. There are two types of osteogenesis imperfecta, type 1 and type 2. Type 2 is lethal, in which the fractures appear in the womb, that is in the uterus, and stillbirth may occur. The bones become so fragile that they show the fractures in their intrauterine life. In type 1 of osteogenesis imperfecta, there is decreased production of alpha or alpha 2 chains, and fractures occur secondary to minor trauma in early infancy. In early infancy, a minor trauma to the child or baby may result in the fracture. This is the radiograph of a baby inside the uterus having spontaneous fractures, multiple fractures you can see, and definitely this baby was born dead. Before starting the elastin, the second most important fibrous protein, I'll try to give you some understanding about the resilience and flexibility. In the connective tissue, the fibroblast cells, they synthesize and secrete collagen fibers and elastin fibers. Elastin fibers, they give the elasticity. When they are stretched, they regain the original shape, and that is called the resilience. But they also need strength, which is provided by collagen fibers. So resilience and strength and flexibility is required in the connective tissue. Elastic or elastin fibers, they provide resilience and stretchability, while the collagen fibers, they provide strength and tear from the overstretching. Elastin, a fibrous protein found in elastic fibers of connective tissue. This elastin is a hydrophobic protein rich in proline, glycine, alanine, valine, and lysine. Some hydroxyprolines are found, but lysines are not hydroxylated, and there is no hydroxylysine. The primary component is tropoelastin. Just like in collagen, it is collagen. Here it is the tropoelastin. Molecule is secreted from the cell. This tropoelastin is wound around another protein called fibrillin. This fibrillin is a glycoprotein, and on which tropoelastin is folded, or you can say that fibrillin is providing a scaffold onto which tropoelastin is deposited, forming insoluble microfibrils. Lysine residues are oxidized and cross-linked to one another, and these lysines, when they are oxidized into their respective aldehydes, they make the cross covalent links, also called the desmosine cross links, which make extensively interconnected rubbery network that can stretch and bend, giving elasticity to the connective tissue. Here is the elastic fiber. The green molecules are of elastin molecules scaffolded on another glycoprotein called fibrillin. Cross links, purple in color, and elastic fibers, they are made from the elastin molecules. These molecules are interconnected via desmosine cross-links. When they are stretched, you see the pattern of these elastin molecules. Here the cross-links made by the oxidized lysine, and when they relax, the stretching force is removed, they resilient and acquire the same shape. Disorders of elastin could be due to poor scaffolding because of absence or less amount of fibrillin 1 protein, and it could be the disorder of elastin degradation. Genetically, when fibrin 1 protein is poorly synthesized, the scaffolding of elastin molecule is not around the fibrin molecule, protein molecule. So a strong microfibril or elastic microfibril is not formed. This is seen in Marfan syndrome due to mutation in the gene for fibrillin 1 protein present in the microfibrils. So microfibrils, they have the poor fibrillin protein having the poor elastic fibril or elastic fiber formation. This is a relatively prevalent inherited disease. Marfan syndrome is an autosomal dominant trait. It affects eyes. Dislocation of lens is there, ectopia lentis. There may be blue sclery. Skeletal systems show patients are tall and exhibit long digits, arachnodactyly, and hyperextensibility of joints. In cardiovascular system, there is a dilatation of ascending aorta with no major clinical manifestation. And this Marfan syndrome was present in Abraham Lincoln, which is the ex-president of USA, and he may have had this condition. The other is the disorder of elastin degradation. Elastin molecules of elastic fibers, they are degraded by elastase. This elastase enzyme is inhibited by another enzyme called alpha-1 antitrypsin. When there is genetic deficiency of alpha-1 antitrypsin, the elastase is not inhibited, causing the destruction and digestion of the elastin molecules. Here in the picture, when the airborne, this is the lining, this is the alveolar space, lung alveolus. These are the flat cells of the alveoli. When the airborne pathogens in the lungs, they cause activation and sensitization of neutrophil molecules. These neutrophil molecules, they produce elastase and other chemical mediators of inflammation, that is called the cytokines. The cytokines and elastase. Elastase in the enzyme, while the cytokines, they are the chemical mediator, chemical molecules which cause inflammation. In the lung alveolus, this neutrophilic release elastase damages the elastic fibers of the alveolar sac. These are elastic fibers. This is the molecule of elastase, and this is elastase inhibitor alpha-1 antitrypsin, just to give you the better understanding. When elastase is produced, it is inhibited by alpha-1 antitrypsin enzyme. When it is genetically deficient, the elastase damages the elastic fibers and degrades the elastin molecules. These destruction of elastic fibers. The alveoli become less elastic, and by passage of time they can be degenerated, producing larger sacs. When smaller alveolar sacs are converted into larger alveolar sacs, the condition is known emphysema. And in the more complicated way, it can change the alveolar tissue into fibrous tissue called the cirrhosis of the lungs. So there could be emphysema, and later on can be converted into cirrhosis due to genetic deficiency of alpha-1 antitrypsin, which is the inhibitor of elastase. Elastase is secreted by sensitized and activated neutrophils in response to airborne pathogens in the lungs. They are activated by immune response and produce elastase, causing emphysema and cirrhosis. However, this condition can be treated by weekly intravenous administration of alpha-1 antitrypsin injection, which can prevent. Two important clinical points I missed from the previous slide. One is that emphysema and fibrosis, the smoking is a risk factor, and chronic smokers, they develop emphysema and ultimately fibrosis of the lung alveoli. Another point is that the emphysematous and fibrosis of alveoli by the elastase could be one of the pathophysiological phenomena taking place in the COVID-19 infection. Coronavirus sensitizes the inflammatory neutrophil cells and which produce elastase, causing the loss of elastin fibers, resulting in emphysema and fibrosis. But this could be the hypothesis and needs approval and from the clinical trials in those patients. And at the moment we cannot say, but it could be the pathophysiology, that is the more and more elastase is produced by sensitized inflammatory neutrophil cells, and with the deficiency, more overproduction of elastase, there is destruction of elastin fibers of alveoli. Now I start with the keratins, two types, alpha and beta. Alpha keratins are found in vertebrates, while beta keratins in reptiles and birds. Alpha keratin is the major protein of hair and fingernails, as well as animal skin. Alpha keratins are rich in cysteine amino acid. These cysteine amino acids, they are sulfur-containing amino acids, and they form the strong covalent disulfide bonds. These strong covalent disulfide bonds provide the strength and provide the stability of the 3D shape of the protein molecule. Structure of alpha keratins. Two helical alpha keratin molecules intertwine, making a dimer. Two dimers twist together to form a molecule of protofibril. A protofibril molecule has 4 alpha keratin molecules. 8 protofibrils combine to make the microfibril. Hundreds of microfibrils are cemented into a macrofibril, as seen in the cortex of the hair shaft. Alpha keratin is hardened by the introduction of disulfide cross-links, making strength and found in fingernails. Here is the picture showing the structure of alpha keratins, which is the self-explanatory. Thank you.